Senhoras e senhores, bom dia. Sejam bem-vindos ao 4º Workshop Internacional, Testes de Diagnóstico com Qualidade Assegurada e Acessíveis para Programas de Saúde Pública. Dando início à mesa redonda, cujo tema é Principais Desafios para os Programas de Saúde Pública Globais, convido como moderadora e coordenadora dessa mesa a senhora diretora adjunta da diretoria do Departamento de DST, AIDS e Hepatites Virais do Ministério da Saúde, doutora ALADDiV Benzaquem. Para falar sobre convergência regulatória, em que direção estamos nos movendo, atualização sobre as iniciativas na Ásia e África, convido a doutora Rosana Pilling. Para falar sobre o papel da OMS, como a pré-qualificação para desempenhar um papel na aceleração do acesso ao diagnóstico in vitro, convido a doutora Anita Santos. E para falar sobre o CDC, como Centro para a Saúde Global, acelerando o acesso a diagnóstico in vitro, convido o Dr. Ron Bala. Com a palavra e a coordenação dos trabalhos, doutora ALADDiV Benzaquem.
Bom dia a todos e a todas. É um enorme prazer estar aqui, apesar da chuva, mas necessária, né, num local tão árido como Brasília. Dizer a vocês que, apesar do atraso, os trabalhos vão iniciar nessa mesa que vai abordar os desafios para os programas de saúde pública. E é um enorme prazer, então, introduzir nesse momento, apresentar a doutora Rosana Pilling, que é minha amiga de longas datas. Nós nos conhecemos desde 2011 ou 2012, já trabalhamos juntos, continuamos trabalhando juntas, mas, assim, a Rosana é uma pessoa maravilhosa de se conviver, mas tem uma carreira bastante ampla. Atualmente, ela é diretora de pesquisa em diagnóstico da London School e diretora do Centro Internacional de Diagnóstico, e na London School também, e ela atuou no TDR, na OMS em Genebra, antes de ir para a London School, e antes disso ela foi co-diretora do Laboratório Nacional de Infecções Sexualmente Transmissíveis no Canadá. Então, é um enorme prazer de ter a doutora Rosana aqui no Brasil, e ela pode começar, então, a sua fala, que é a convergência regulatória, em que direção estamos nos movendo, que é um tema que, atualmente, ela se dedica nesse Centro Internacional de Diagnóstico.
Good morning. Thank you very much, Adele, for the kind introduction. So this morning, what I'm going to do is just go over the work that we've been doing for the last 2.5 years on regulatory convergence. I remember the first time that the Gates Foundation and Grand Challenges Canada approached me about doing work on regulatory issues to do with diagnostics. I said to them, we did a survey when I was in WHO about how countries regulate diagnostics, but I'm not a regulator, nor have I worked in industry, so I really don't know very much. And they said, that's good, because you're a neutral party. You could look at, with fresh eyes, what's happening and make some recommendations on how to go forward. So starting from 2012, we started to look at what are the top challenges in terms of regulation of in vitro diagnostics. And what we found when we looked at how countries regulate in vitro diagnostics, and I'm talking about outside of blood banking diagnostics, so this is diagnostics for infectious diseases in general. There's a huge variety of ways that countries regulate diagnostics, mostly not regulating diagnostics very well. The approval processes seem to be very lengthy, and especially for imported tests, as long as 5 years to get it through. Now, considering that most diagnostic tests have a generation time of about 5 to 7 years, this is a long time to get approval in order to make the returns for investment for companies. And a lot of regulatory approval processes are not transparent, meaning that you cannot find information on the web about what the approval process is. So as many companies know, you move into a country where you don't even speak the language, you have to take on an in-country partner, but there's nowhere that lays out what the process is. And you just keep paying money, you keep submitting documents, you have no idea when you would get approval. And that's a scenario in some countries. The reviews often lack quality standards because unlike drugs, there isn't an international ICH framework, etc. And we found it in many of the studies published, and also the review standards are not there for in vitro diagnostics, especially clinical performance studies. I've had companies say to me that their test is 100% sensitive, 100% specific, but it was only done in 3 patients that are positive for that disease, and that's why they get 100% for both. And some of them said, oh, it's done in like 15 patients, as though that is really good enough. And so I think these kinds of standards need to be made a lot more transparent so that people know how to approve a good test. And so as a consequence of these 5 top challenges, tests are often sold and used in the developing world without much evidence that they exist. For example, the fake Ebola test that we saw in Nigeria as soon as the first case of Ebola was announced in Nigeria, that is really very scary in terms of public health control. We often have the long and very, very difficult regulatory approval process, the cost of doing that passed on to the end users so that it makes the final product unaffordable. And also, because of the complicated approval process, companies with good quality tests just are really unable to or unwilling to compete in the market flooded with bad tests. And also, companies really don't want to target small markets because it's not worth their while. So we have this very difficult scenario with smaller countries not having access to the tests they needed. And there's been a lot of work done by the Global Harmonization Task Force, which was formed in 1992, but there hasn't been, apart from the EU, there hasn't been a lot of standardization. So if we look at the diagnostic pathway from having identify a really good target, a promising target, and then putting it onto a platform, doing proof of concept studies. For a new platform, that's already about 10 years. And then you still have to go through clinical trials and regulatory approval anywhere between 2 to 5 years. And then policy and uptake sometimes could take a long time. My guess and my experience has been 5 to 7 years, but quite often the UK Department of International Development told me that it's as much as 10 to 14 years when they first see a study published on an intervention before countries actually adopt it. So end-to-end is about 20 years and a huge valley of death, with a lot of promising tests never making it to the end. So we started in Africa with just trying to get people who are normally regulating drugs to have them build capacity on how do you look at a new diagnostic test. So we told them that the laboratory-based evaluations would give them analytical sensitivity and specificity, and you could also look at reproducibility of an assay under laboratory conditions. So this would give you the best that the test is capable of doing in the best hands, in the best environment. And then for point-of-care tests, you quite often have to take it out in the field and see how it performs in the field, because it's meant to be done by field workers. And for that, you get clinical performance and also how rugged it is. Can it survive the high heat, high humidity, and dust? And you also can look at feasibility of introducing it under field conditions. And those are field performance studies, and it gives you clinical performance data. Now, the part that the regulation is not concerned with is all the utility studies, the cost effectiveness, the implementation research, and estimating potential impact. Those are for departments of health and control programs to do. So we need to make sure that the regulators don't also try to do that as well. So this is the diagram that we use for the teaching of the regulators on what roles do all the players play. And since Anita is speaking after me, so I'd like to just point out that for the pre-qualification of diagnostics at WHO, there's a dossier review, but there's also an inspection of the quality of the manufacturing process, as well as the laboratory evaluation. And that constitutes the pre-qualification. And quite a lot of people, including ourselves, as well as a lot of academic and NGO groups and CDC, do field trials for clinical performance studies. And then once diagnostics is approved, then there will be program issues in terms of looking at where to place them, and then policy development, getting financing, which is a huge issue in terms of diagnostics, and then finally is use in programs. Now, so for our task that we were asked by the funders to do is to look at what the GHTF had done. The founding members of the GHTF are the EU, US, Canada, Australia, and Japan. And it started out to try and look at how you could harmonize regulation of medical devices, which includes in vitro diagnostics, to ensure the safety and effectiveness of the quality of medical devices. And they've actually got several study groups that have produced a lot of documents on which everybody, the foundation for regulating medical devices in vitro diagnostics are based. And so we still find that these are very useful documents. And for the purpose of regional harmonization and global harmonization, these are very important documents. And as you'll hear later, in 2012, the GHTF transitioned to a purely regulatory body called the International Medical Devices Regulatory Forum, IMDRF, whereas the GHTF is actually a platform for both industry and regulators to interact. And so the nature of the body has changed, but nevertheless, IMDRF still maintains all these documents on their website. Now, the Asian Harmonization Working Party, which was started in Singapore several years after after GHTF, has now got 23 member economies. And you'll notice that there are members outside of Asia, so Middle Eastern countries, Chile, and South Africa are in the Asian Harmonization Working Party, and I think Tanzania is wanting to join the AHWP, and their case will be reviewed at the annual meeting in Korea 2 weeks from now. What the Asian Harmonization Working Party had done was to actually use the GHTF guidance and elaborate documents on how to help countries with using the GHTF principles to develop guidance for their own work. And so there are 6 working groups, and the in vitro diagnostics come under working group 1, which is for pre-market submission and CSDT. And especially working group 1A is dealing with in vitro diagnostic devices. We also have a group which is working Group 2, post-marketing surveillance and vigilance, and that is very important to diagnostics, and we work with that group as well. What the AHWP had done is really develop a lot of documents, including an IVD regulatory framework, because they do feel that there is a difference between IVDs and medical devices in general. And so after years of trying to work within the medical devices group, there's now an independent IVD regulatory framework document which can be adopted by all different countries. Now what we did was we started in 2012. In March of 2012, there's an East African Community Medicine Harmonization meeting. And with that, a member from that committee actually appeared in my office in London in April of 2012 and asked about having some means of looking at whether it's possible to harmonize IVDs. And so we worked very hard, and in July of that year, I'm sorry, this it's not showing up very well. In July of 2012, we had the very, very first group, regional meeting in Nairobi. And rather than just the 5 countries in the East African community, which is leading the way in terms of regulatory harmonization, South Africa, Nigeria, and Ethiopia wanted to join. And so we had 8 countries at that meeting in Nairobi. And everyone was really committed towards trying to find ways to streamline as well as to harmonize approval processes for tests that are urgently needed, such as HIV point-of-care tests for CD4 viral load and early infant diagnosis, as I had talked about in my presentation last night. Those are really urgently needed to save lives, and so everybody's committed. At that meeting, we actually made a tentative program for a Pan-African harmonization working party. And in December of that year, at the meeting of the African Society for Laboratory Medicine in Cape Town, we officially announced the formation of the PAHWP in December. But in the meantime, between July and December, we were funded also by the German International Corporation, GIZ, to do a lot of landscaping of what's being done about the approval of diagnostics in different countries in Africa. So then, after we established the PAHWP with a framework, we started to look at how we could collaborate with different regions. And especially in Latin America, we started talking about a Latin America alliance for in vitro diagnostics at a meeting at an advanced course in diagnostics that we hold every year in France. And a group of people who are all still here, including Carlos and Adele. Yeah, it's a good. Yes, Freddie. Yes, there was the core group that started in September of 2012 to look at how this can be done in Latin America. And so you could see here that we've been really busy having a whole lot of different meetings to try and figure out how to utilize all the documents that are in existence and then identify gaps. In particular, I'm very pleased to say not only have we made a lot of headway in terms of the African harmonization, because at our launch in December of 2012, 15 countries in the SADC bloc, the South African Development Community, also wanted to join. So, and then Sierra Leone and Gambia and Ghana also wanted to join. So totally in Africa, we have now 23 countries committed to harmonization. And what we've done is to have these interregional harmonization working parties so that we could learn from each other, as well as to help with the development of shared learning materials. We also approach ISO about putting everything that we've done into ISO standards for IVDs, and that's been really well received. And so that we know that it could be sustainable. For post-marketing surveillance, we have the collaboration of the African Society for Lab Medicine to be able to use their accredited labs to do post-marketing surveillance. So in December and November of this year, we will have our Pan-African Harmonization Working Party have its third international forum in Cape Town, and we will be also doing it together with the African Society for Lab Medicine meeting in Cape Town. Now, so what we've done was we started with the countries in Asia, and then in 12 countries in Latin America joined in 2012. And then we have the Pan-African group, starting with the 8 countries to begin with, and now totally we have 20 countries, and then 3 more had joined later. So all in all, we have a lot of momentum, a lot of commitment, and we really need to make it happen now. So in terms of the goal, we wanted the goal to be very clear, very simple. The goal is faster access to affordable, quality-assured point-of-care diagnostics for the moment, but it's actually for all in vitro diagnostics. And we want to also, in a lot of countries, prevent low-quality diagnostics from even entering the market to waste resources. The strategy is to define a pathway that is better, faster, safer, and cheaper. And these are the priorities that we have set for harmonization. These are what we think of as universal standards: risk classification of IVDs, a common registration file, and we're using the WHO pre-qualification sample dossier for CD4 as a trial common registration file. Because when you really think about it, you know, for a product, why would you have to submit different types of dossier to different countries? It should be the same. And because the essential set of information for review should be the same. Quality management audits, we're hoping that the MD, the single audit program by IMDRF could be used. Clinical performance studies for new diagnostics, we're doing a lot of work to help with the e-learning for that. And then post-marketing surveillance, Anita is leading this effort from WHO, and we also, for other diseases, we have a post-marketing surveillance network as well. So the Pan-African Harmonization Working Party, we now have the chair of the Working Party is the EAC, and there's a rotating chair. Right now is Kenya. It's Uganda, and next year will be Tanzania. The vice chair is Nigeria with NAFDAC, and secretary is South Africa. And South Africa is making a new health products directorate, which will be focused on medical devices and IVDs. And all this is housed under the African Union, NEPAD agency, the new Partners for Africa's Development. And so this is a very busy slide. I just want to show you that with all these harmonization priorities that I've already told you about, the result is we want more streamlined regulatory processes for approval. We want to reduce duplication in the number of clinical performance studies companies have to do in every single country that they seek a registration. And we want to be able to save companies time and money in terms of audits and clinical performance studies, so that the savings could be passed on to the customers. And so we want more affordable IVDs with faster access to quality diagnostics and preventing bad ones from entering the market, with the outcome of better patient outcomes, and also for regulation to support innovation, to go hand in hand. We used to say we mustn't let regulation be a barrier to innovation, but what we want to really say is that regulation should go hand in hand with innovation from industry as well as from public sector. The post-marketing surveillance would then create more public confidence in diagnostics. Now, this is a very busy table, but I want to show you that the essential documents on how to set up an IVD regulatory framework are there from the GHTF. There's an IVD framework, there is definitions, and also a lot of this can already be adopted by all the regions without any additional work. But there are a number of documents that would need a little bit more work on the IVD in particular. And in Africa, we are actually piloting a joint review of clinical performance data on specific commodities that are priority HIV point-of-care tests. For example, we had done 2 workshops in Africa in the last year to teach regulators from 10 countries in Africa how to review clinical performance studies. And then the first one was a basic course, and then we also did a more advanced course just 2 weeks ago in Dar es Salaam for the 8 countries, because 2 West African countries cannot join us. And to take the regulators from just understanding the basics on sensitivity, specificity, etc., to understanding how to compare quantitative assays, what kind of information you need to know how to compare one quantitative assay against another. And so we are now poised for these regulators from the 8 countries to now really review real dossiers, and we've been talking with Willie Urasa from WHO pre-qualification that next what we will do is to review the dossier of the 2 pre-qualified point-of-care CD4 tests, and so that all the countries can review the same set of data and be able to give their approval in country without further studies in their own country. And so there's a lot of work happening also with post-marketing surveillance, and for the audits, we're hoping that everybody can eventually just accept the single audit program. I think there'll be more talk about this later, so I would skip this. So this is where our advisory group is under the African Union, and these are our working groups. And I really am very pleased to say that in May of this year, the EAC jointly approved 3 antimalarials and 2 health products as a community. And when I asked if any of the data on these studies are from the EAC countries, in fact, none. So this issue of every country insisting on data from their own country, I hope, is now a thing of the past. As long as the studies are done well, up to international standards, regulators should be able to review the data and be able to approve the product. So EAC is very keen for the next set of products to be approved as diagnostics. And so now back to our pathway. We're hoping that we could really, streamline the novel diagnostics for priority for HIV at the moment. We also need to do that for Ebola and other tests that are important in global health security. And so we really need to look at what we're doing in terms of regulation and balance that with the risk and the benefit of saving lives. And so it really need to be really educating the regulators on the risk-benefit ratio in terms of approving tests, and not to ask for the perfect test. And so we've been funded by UNITAID to try and accelerate the evaluation, the policy development, and set up quality assurance tests for point-of-care tests for HIV. And this goes hand in hand with our regulatory work. And so hopefully that we will be able to have tests that are really approved very quickly. We also set up 13 sites through an open call for sites that are expert sites at evaluating these point-of-care tests for HIV. And each site not only have the gold standard and they're proficient at the gold standard, we look at the proficiency records because each one has to subscribe to an international proficiency program for CD4 and viral load and early infant diagnosis. We also have generic protocols that standardize the evaluation across all the sites so that we could aggregate the data, especially for early infant diagnosis that's often necessary. We aim to have these evaluations done between 6 to 9 months. For CD4, it may even be shorter than that. And this, every 2 or 3 months, we will review data from these trials with the regulators from different countries for approval. And each one of these trial sites have already got IRB approval for going forward with the evaluation, so that when the product is ready, can immediately go into evaluation instead of waiting for another 9 to 12 months to get approval. So this is how this is our mechanism for trying to accelerate and raise the standard of evaluations for new tests. So this is just a slide about the EAC approval of the new medicines, how together with the WHO pre-qualification program, it has really streamlined and shortened the time to approval, and yet maintaining very, very high standards. So this is really very good news for us. So moving forward, there's a lot of things that are happening. We had an inter-regional meeting where AHWP and the African Harmonization Working Party agreed to collaborate, and we have ISO standards being developed for IVDs. And the AHWP has actually developed a playbook, a play-by-play on how to set up an IVD regulatory system, which can be used by all countries. You will hear more about what's being done in Latin America with Brazil championing a common audit initiative. We also learned from the APEC group, the Asian Pacific group, that they have created a virtual campus for e-learning and a community of practice for regulators, for IVDs and medical devices, which is really good because we also have developed e-learning material on how to review clinical performance studies that could be put onto these virtual e-learning modules. And we really, now moving forward, need to think about how to brainstorm on new models of public-private collaboration to work with industry so that regulation and innovation can go hand in hand. And we need countries to look at technology assessment, strategic placement of new technologies, and build quality assurance systems to ensure that we will have the kind of impact that new diagnostics can have. So I'd like to give my thanks to all the people who work with me at the London School, as well as all our partners. We have many partners that work with us that give us a lot of input, and some of them are here, CDC, CHI, as well as UNITAID that funded us, and also the WHO as our partner for all the pre-qualification work that they've done over the years, and all the countries that are really very, very helpful in terms of their commitment. Thank you.
Thank you, Rosana. Nós poderíamos passar na sequência para o próximo apresentador, Anita Santos, que ela é oficial técnica do Departamento de Medicamentos Essenciais e Produtos da Saúde da OMS em Genebra, e também ela trabalha no programa de pré-qualificação de diagnóstico in vitro e responsável pela vigilância pós-mercado na OMS. Então, ela vai nos brindar com a conferência sobre o papel da OMS, como a pré-qualificação pode desempenhar um papel na aceleração do acesso ao diagnóstico in vitro. Doutora Anita, por favor.
Okay, so this morning I'm going to talk to you a little bit about what WHO pre-qualification is. You've heard many people talk about it. Perhaps you've heard about it in conferences, and I'm going to give you just a very quick run through about what it is and what its scope is and what our mandate is, and also to talk to you a little bit about a new piece of work on post-market surveillance for IVDs. So basically, when we're talking about pre-qualification of IVDs, what the aim of the whole program is within WHO is to facilitate access to safe, appropriate and affordable IVDs that are specifically needed for resource-limited settings. So that really does limit a little bit the scope of what we do is to those sort of diseases that are most prevalent in resource-limited settings, that are products that often don't undergo regulatory review elsewhere. So our priority products are HIV, malaria, hepatitis C, hepatitis B, and other HIV-related diagnostics such as CD4 and viral load. And we're most interested in looking at the sorts of formats of IVDs that are useful are appropriate for resource-limited settings. So preferably capillary whole blood, preferably no need for cold chain, preferably as robust as possible, preferably a smaller instrument or no instrument as possible. So we don't actually look at the scope of all IVDs that exist. We very much have a narrow scope on the sort of products that countries in particularly sub-Saharan Africa, where there is a lack of regulation, where they're unable to actually review the products themselves. So we're able to provide findings from the pre-qualification program that give you independent technical information on the safety, the performance and the quality of IVDs. And a number of UN agencies, WHO as well, member states and other procurement agencies and implementing agencies actually use pre-qualification findings to help make procurement decisions. We've just entered into, or we're currently still entering into an agreement with CDC around having a joint assessment mechanism. There is also a similar sort of mechanism to pre-qualification within CDC, particularly for HIV rapid tests called the USAID waiver list. And we're working together to try to find a way to harmonise and for each partner to do a little bit of the assessment. And that means that rather than the manufacturers having to go to each separate organisation, that they'll just be one conduit, and then there'd be a joint list. So that's quite an exciting piece of work that we're just working with our lawyers on the last piece of making that formally available. So in terms of what is pre-qualification, it's a very standardised procedure, irrespective of the actual IVD or the type of product. We have a very standard procedure for the administrative aspects as well as for the technical and review aspects. And we actually, it's comprised of three main areas: a review of a product dossier, a laboratory evaluation that's performed by WHO, so it's independent of the manufacturer, to evaluate not only performance but also operational characteristics. And this is the sort of information that policymakers and program managers might need to help guide product selection decisions, and also an inspection of the manufacturing site or sites. If there's a key supplier involved that makes a very key part of the product, we might also need to go and visit them. So I'm just going to go through each of these 3 steps and the pre-submission step, which is really the 4th step, in a little bit more detail. We have redesigned the pre-qualification process a little bit this year. We're calling it streamlining. We're trying to streamline, making it a bit faster, more efficient and much more transparent. And so we actually have quite a large emphasis on the pre-submission stage, which is really working with the companies to make sure that the IVD and the site are ready for pre-qualification. We looked a little bit at the way in which FDA works, and we were wondering why their timelines to a PMA were so short, and we realised it's because they put a lot more effort into the pre-submission than perhaps we had. And so the first step is for a company that has a product is to submit a pre-submission form. So it's a very standardised tick-a-box form where you say who you are, what your product is, and give us some basic sorts of information that allows us to sort of understand how ready the product would be for pre-qualification. So, for example, we ask about what regulatory approvals do you have. We ask about any clinical evidence that has been, any clinical trials. So we're just asking, have you completed clinical trials? We don't ask for the information until later in the dossier, but we're trying to already get a feel for how ready you are for pre-qualification. So once we have this information, we actually have to decide, is it a priority product or not? So, for instance, if it's a product for syphilis, unfortunately at the moment a syphilis sole test is not currently one of our priority products, although a HIV syphilis test is, funnily enough. And so if it's a syphilis only product, unfortunately at this time we're unable to look at to take the product through pre-qualification, however we keep it on our files. So then we actually we do have a fee structure, so we're very grateful for the support of our 2 donors, Unitaid and the Gates Foundation. However, we're also grateful for the support that manufacturers give. There is a fee, which is what we consider the support, and also the supply of the reagents, and we do gratefully acknowledge the role that manufacturers have in paying the pre-qualification fee. It's $12,000 in total. There's a $4,000 screening fee, and then if the dossier is deemed acceptable and complete to go through, the additional $8,000 is charged. So once we receive the product dossier, we actually screen the dossier to see if everything is in there. As Rosanna mentioned, we do have a sample product dossier which we put together for a fictitious point-of-care CD4 device. However, it could be applied to any other type of product. We do follow the GHTF STED documentation, the summary technical documentation, I have to remember, for essential principles, and we know that that's in the midst of being revised at the moment, but we were able to use the document that's currently in practice. And so what that means is that a dossier that is put together for WHO pre-qualification purposes should contain the same sort of information that you would need to submit to other regulators who are subscribing to GHTF or IMDRF guidance. Often maybe it might be in a different order, and so we have a table of contents, and if the order is different, you can just tell us where that piece of the dossier is. We allow only 2 rounds of supplements to be submitted. In the past, we had dossiers that were incomplete, and we had up to 7 or 8 rounds of additional information submitted to try to complete the dossier. We have to outsource the dossier assessment to an external expert, so obviously we're only interested to send it once it's complete. And we found that this was leading to a bit of a longer timeline, and so we've actually reduced that just to 2 rounds. And obviously we're working with the manufacturers and telephone conferences and emails to get a better understanding about when data is ready. So in terms of the dossier itself, we're down here on the bottom left there. As I said, we wait until the dossier is full and complete. Administratively, we look through and see for Section 7 on clinical evidence, is there one manufacturer-led clinical trial, is there one independent clinical trial, is there stability data, etc. It's sent to an assessor, and they actually rate the dossier based on a rating system of non-conformities. This is also something new we've just introduced this year, and it very much aligns with the way in which we rate non-conformities in the inspection process. And again, this is based on GHTF guidance about how to rate non-conformities. So it goes from level 1 through to level 5. A level 5 is what used to be called a critical non-conformity, and if you have a level 5, that would lead to an immediate closure of the pre-qualification assessment. What that means is that there really was some evidence in the dossier that indicated to us that we really shouldn't proceed with the pre-qualification assessment, and we'd work with the manufacturer and tell them what that piece of evidence was that was non-conforming, and try to find a way for them to generate that evidence and to come back into the cycle. So once the dossier is reviewed and any non-conformities are rated, you have level 4, level 3, level 2, we actually ask the manufacturer to submit an action plan, a corrective action plan, to say how they'll go about generating that additional evidence. It might be that they didn't understand what we needed, or it might be that there needs to be a new study performed, and that is all very acceptable as long as you tell us how you're going to go about doing it and the timelines. And so obviously there might be missing pieces of information you need to submit afterwards. These are what we call amendments, and so we obviously have a step where the same assessor reviews those amendments. So the dossier is actually quite a lengthy process. It's probably the thing we start first and the thing that is finished last. There is a lot of data in there, but this is actually really, if you have ISO 13485, you're obliged to have a technical file and a design file that describes how you design your product, how you manufacture your product, how you distribute your product. So this is data that really you should have. And obviously for HIV products, these are normally very highly regulated, and we would really, this is an expectation. For site inspection, we've also slightly changed the way that we're doing this. We found that we were very often going on site inspection a bit too early, and that sites were not really ready for us to be there on site, and we were finding quite a lot of level 5 non-conformities. In the old system, we gave the companies the opportunity to spend another year, 2 years, build a new factory, get a consultant in, and then we'd go back and see how things are. And obviously that was really leading to a long time to pre-qual. So we have a new way of doing it in which we stage this, what we call readiness for inspection. The first stage is normally a desk audit where our inspector asks for certain quality management information, so the quality manual and certain documents around supplier control, QC, some of these key elements that we found were consistently not done so well, that gave us a real insight into how the product was really being manufactured. Also, we have a lot of assembly of components, so that a critical part of the device might be made elsewhere, and either as an actual strip or as a sheet of nitrocellulose with antigen, or even actually already in a device for relabeling. So we found there was not always all the manufacturing was done in one site. So this is to sort of make sure that we have a very good understanding of those manufacturing flows. And then the stage 2 would actually be the on-site inspection of the quality management system. And we found this to be much more effective in terms of the use of our resources. The cost of an on-site inspection is about $50,000. We normally take 3 to 4 inspectors. We're there 3 to 4 days. So as you can imagine, flying people from all over the world, we have a whole pool of consultants who work as inspectors and flying to these different parts of the world. So we do want to reserve that on-site inspection for when sites are really very ready for us. And then we follow the same system of non-conformities are rated. In our new streamlined approach, any level 5, which is a critical non-conformity, would lead to immediate closure of the pre-qualification assessment. However, there are normally always 1 or 2 little things noted, and again, we ask the manufacturer to submit an action plan to tell us how they're going to correct and prevent things in the future, and we may elect to do a follow-up inspection. Now, these can either be by a desk review of documentation, or on site. And we really do try to take a risk-managed approach to this, to have as few times on site. We realise that's a burden also on manufacturers to always be preparing for inspection. They have FDA, notified body, they have us, they might have any number of people, and so we're trying to actually make sure we minimise the resources that they need to expend. And for the laboratory evaluation, this is actually really the foundation of our whole program. We've been performing laboratory evaluations for over 25 years. And until about 7 or 8 years ago, the results of a WHO laboratory evaluation was actually what determined if you got onto the WHO procurement list. This concept of pre-qualification actually is an alignment with what our medicines and our vaccines colleagues do already. And so this adding of the dossier inspection stage only happened, fairly recently. But the laboratory evaluation, as Rosanna mentioned quite nicely in her little figure, it really is a very top-level laboratory-based evaluation. So it's not replacing an evaluation in the intended setting, which is normally the field. It is carried out in WHO collaborating centres, so it's normally in a fairly well-performing laboratory. But we do have a standardised evaluation protocol that they follow. We have a predefined acceptance criteria, so for this one it's very clear, you pass or you fail. And we actually do publish all of the data, irrespective of if the product meets the acceptance criteria or not. The reason we do this is that we're not really acting as a pure pre-market assessment, even though we call it pre-qualification. We're aware that a lot of products may be out there, they may be being used, and so having this independent technical data is very useful for countries who may have elected to use a product before it was pre-qualified. So in terms of once we have these 3 critical pieces finished, the dossier, the site inspection, laboratory evaluation, we need to make a PQ decision. And I've kind of already alluded to it, but we do certainly look at the outcomes of the dossier assessment and the acceptance of the action plan. The same for inspections. There must not be any level 5 non-conformities, and you must meet the acceptance criteria for laboratory evaluation. Once you meet all of those, the product is pre-qualified. We put together a summary public report, so we don't actually make public the findings of the dossier and the findings of the inspection. This is between ourselves and the manufacturer. We actually have a confidentiality agreement, so we keep those aspects of pre-qualification within confidence, because we feel that sometimes the information that's contained is very difficult for a lay user to digest. And so we have a very summarised report that is published on our website, and then the product becomes eligible for WHO and therefore UN procurement. And within WHO, we actually go through a process of signing an LTA, a long-term agreement, each year with manufacturers of pre-qualified products, and that assures that you're on our catalogue and therefore on UNICEF catalogue. And also Global Fund. And at that point we actually do negotiate a price per test or a price for the instrument or reagents, but that's not a volume-bound agreement. So we call that sort of a high ceiling price, and then countries are free to use that to negotiate even further if they wish. So I've talked in quite a lot of detail about what we call the full pre-qualification assessment. We also have introduced an abbreviated PQ assessment. We had a similar mechanism in the past. It was called a fast track, but it was very difficult for us to implement. It was if a product had undergone stringent regulatory review somewhere else and they submitted it to WHO, that we would fast track parts of the assessment. However, out of the 250 applications we received, there was probably only about 3 or 4% that were actually eligible for this kind of assessment. So we've actually expanded the scope of what we're now calling abbreviated PQ. People used to think fast track meant you could jump to the head of the queue. We said, no, it doesn't mean that you jump ahead of others. It just means that we abbreviate certain pieces. So the name change is quite symbolic there. So we really have 3 categories of products that get submitted to PQ. As I said, this very rare occasion where the product that's submitted to us has already been stringently assessed. We have a scenario 2 where the product that's submitted to PQ hasn't been stringently assessed, but there exists a regulatory version that has been. And so this is one of those nuances of the IVD world, is different regulatory versions of what appear to be, the same product, the same HIV RDT. And I've had many conversations with our medicines colleagues and trying to have them to understand this. They still find it a very hard concept to get their head around, and what we say is, different regulatory jurisdictions have different requirements, and hence you have this splitting off of different regulatory versions. And And of course we have this third scenario where the product submitted to us has never been stringently assessed, and most likely the company has very little regulatory expertise. And I just want to point out the most important part about what we mean when we say stringent assessment. People very often say to me stringent NRAs, and I say for diagnostics it doesn't really work well as a term because NRAs use a risk-managed approach for regulation of IVDs. We can't assess and regulate all IVDs to the exact same high stringency. So obviously we take HIV products, those that are used in the blood screening, and regulate those with more stringency. So we consider for CE mark it's a List A Annex 2. For FDA it's a PMA or a BLA, so a 510K is not considered as stringent. Health Canada and the TGA it's Class 4, and for Japan it's what's called the ministerial approval. And just to sort of, I'm going to try to skip through these slides a little bit quickly, but the main difference with the abbreviated PQ procedure is that if you're eligible for abbreviated, you do not need to submit a dossier to WHO to review. That's the main difference. And the site inspection is much briefer. It's fewer inspectors and fewer days on site. We still do the laboratory evaluation. So this is just to describe a little bit about if it has been stringently assessed. As I said, no dossier is required, a shorter inspection, lab evaluation. For the scenario 2, this is where it gets a little bit tricky. So we very often have a company that may have many different regulatory versions, and it's often very difficult for us to understand the differences between the regulatory versions. Sometimes it may just be in the QC lot release, sometimes it may just be labelling, it may be the IFU, but in other circumstances it's actually a completely different manufacturing site. So we now have an annex that for the pre-submission form that if a company discloses that they would like to be abbreviated, we ask them to fill in where we can look at the key differences, and we actually make a decision about if there are substantial differences between the products or not. So if there are substantial differences, we do the usual PQ, and if the differences are not substantial, so things like labelling an IFU, like they're often very country-specific or regulatory-specific terms, we would be able to follow the abbreviated assessment. And obviously the third scenario would just be the very usual PQ assessment, and this is actually the scenario that we have to use the most of. So what happens after the product is pre-qualified? We receive a lot of funding for pre-qualification. There's less funding out there for countries to have a post-pre-qualification or a post-market arm to what they do. So for us, we consider it an activity that includes both reactive, so once a problem has occurred, and proactive, trying to identify problems before they happen. So reactive is obviously through complaint reporting. You see a problem, you make a complaint. Proactive is obviously through lot testing, so testing samples of products that have been delivered to country or even delivered to testing sites. We also have a mandatory system for manufacturers to notify us of changes to products. And this is where in pre-qualification we also don't want to stymie innovation. We want to be able to have manufacturers to continue to improve their products, to be able to get better suppliers, maybe cheaper suppliers, improve manufacturing processes through automation, make improvements to the product. And so we have a very distinct change notification procedure, and I'm going to talk about these a bit in turn. So post-market surveillance is a subject very dear to my heart. We're currently writing normative guidance. We had a meeting 2 weeks ago. Some people from the audience were actually there. And we're going to present a fairly final draft at the ASLM conference in Cape Town at the start of December for public comment. And this is talking about complaint reporting, field safety corrective action, and therefore field safety notices in the reactive arm, and the proactive lot verification. I'm just going to skip through this one, but I think with post-market surveillance, the real point of difference is there are many stakeholders that are involved. We have end users and implementing partners that must actually identify problems and report them, and that's sometimes very difficult. We have national reference laboratories that might be performing lot testing and might identify issues. We have manufacturers who normally have quite an obligation to do post-market surveillance but find it very hard to get information from the field about performance of their test other than very anecdotal information. We have NRAs in some areas of the world that actually have a lack of capacity, just a lack of people who are working on IVDs, let alone on post-market surveillance. And so WHO is sort of stepping in a little bit to bridge the gap until we can improve that capacity. So we're promoting this practice of post-market surveillance, making sure that manufacturers are investigating problems and complaints when they come to them and taking action if required. And so in terms of talking about changes, so that's all I'm going to talk about post-market surveillance. I've got a whole nother presentation if people are interested. But in terms of once the product is pre-qualified, as I mentioned, it might be necessary to implement changes to your product to improve it. They may be a minor change, they may be a substantial change, or even constitute what would be a new product and therefore a new regulatory submission. So as part of a manufacturer's quality management system, they should have a system in place that designs, validates and implements changes and determines their significance. Again, it's all about risk, you know. When you make a change, do you raise the acceptable risk? I might move on to this because this is actually really the sort of, so we have a guidance document on our website that talks about what we consider to be a minor, a substantial or a new product. And minor changes are often things like typographical errors and grammar and just making instructions for use more readable. Very often the manufacturers who produce tests, English might not be their first language, and certainly the users' first language may not be English. We have a lot of translation of IFUs, so very often this is what we call a minor change. Substantial change may be change to the QC procedure or change in automation in the facility. A new product would probably be something like a total change in antigen supplier where the performance characteristics of the product are really altered. We have a lot more information in the actual procedure document on our website. This is a very brief flyover. So we do have a lot of new guidance that's come out this year. Rosanna mentioned the sample product dossier. We also are looking to write a lot more guidance documents on specific aspects in the dossier which we've seen to be poorly done. Stability, obviously the clinical evidence piece, and some of the analytical work as well. We've also updated our information for manufacturers on inspections because as I just said, we've changed it quite a lot. We have this staged approach. The change notification form and the accompanying guidance, which talks to this previous slide, and of course the forthcoming post-market surveillance. I must say also the post-market surveillance is very much based on GHTF guidance and the MedDev guidance. MedDev is a sort of an accompanying document that helps put it into in practice in the European Union, and there's a number of reporting forms. And so again, it's to sort of make sure that we have harmonised requirements and what we would expect of WHO pre-qualified products is the same if it was sold in the European Union. And just two last slides actually, but just to talk about, I've sort of talked a lot about what pre-qualification is, but the title of the talk was about how is this increasing access. Now, we do hope that by being on a procurement list and by having WHO talking a lot about the value of pre-qualification and the way in which countries can use the data, that in a way accelerates access. But we're also undertaking a new activity that takes interested NRAs and encourages them to fast-track the national registration of IVDs that have already been assessed and approved by PQ. Now, our medicines group already has an activity and has had this in place for 2 or 3 years now, and Rosanna mentioned a little bit about some of the findings with the 3 antimalarials and the 2 reproductive health commodities that have already been fast-tracked through this mechanism. So what it would be is an exchange of the critical information that we find in the pre-qualification assessment on safety and quality performance, and it allows for the pre-qualification assessment to be used by NRAs without compromising national autonomy and within the bounds of confidentiality. And the manufacturer is very much a part of this process, and in fact they are the ones that approach WHO to say, I want to register my product in country X, I'd like you to facilitate it. We have our first meeting with IVD regulators in Kenya in the first week of December, overlapping with the Cape Town conference, but we're hoping to have good outcomes. And we have a number of ways you can contact us. You can either contact me personally, but we also have a WHO diagnostics email address. We also have a mailing list, so whenever we update documents or we have new events, it's a really great way to find out information rather than trawling through our website, which is not always the easiest. However, we have made some strides to try to improve its usability, and you have it there. So thank you very much.
Thank you, Anita. And now it's you, Ron. So, o Ron Ballard, ele é diretor associado para a ciência de laboratório do Centro de Saúde Global do CDC Atlanta, nos Estados Unidos. E, como eu dizia, talvez ele não saiba, mas ele foi quem nos ensinou a utilização do DTS, que é um método de avaliação externa da qualidade, incorporada também pelo Ministério da Saúde do Brasil na questão da avaliação da performance da execução dos testes rápidos por profissionais de saúde. Então, com vocês, o Dr. Ron Ballard.
Thank you very much, Adele. Well, thank you, and once again, thank you for inviting me to talk to you today. The total title of my talk is CDC as a Center for Global Health, Accelerating Access to In Vitro Diagnostic Tests. We're rather different to either the London School Department and also WHO in that we're not a regulatory agency. With two notable exceptions. One is our malaria lab acts as if you like, the gatekeeper for diagnostics for the President's malaria initiative. In other words, selects the best rapid malaria tests for that initiative. And also one of our HIV labs actually acts as the gatekeeper for HIV diagnostics for PEPFAR, the President's emergency program for AIDS relief. So we're not a regulatory agency, but we have an enormously wide range of subject matter expertise within the agency, and you'll see what our global reach is like in a minute. We have state-of-the-art laboratories that can perform laboratory evaluations, and we have field sites scattered around the world where we see lots of clinical disease. So we perform surveillance worldwide, not only to inform the global community, but also to inform the US community of what's literally one plane ride away. But also, it's important to recognize that we perform applied rather than basic research. So, in other words, we don't tend to compete with NIH. We look very much at detection, typing, markers for antimicrobial resistance in order to help inform public health programs. We have an enormous access to clinical samples, as you can imagine, and so our laboratories can perform these in vitro evaluations in our laboratories, but also we send out panels to laboratories around the world for proficiency testing. We are encouraged to work with the private sector, and we have many cooperative agreements with commercial partners as a result. And I'll give you some examples of that later on. And we're one of the US agencies that provide small business grants for specific projects. So if we decide that there is a gap that needs to be filled, we send out a call for applications for funding for those specific projects. So this is a bit of it looks as if somebody's been ill on this slide actually, but literally it gives you an idea of the kind of reach that we have in terms of where our staff are and around the world, and you can see that we have more than 1,600 staff located in 50 countries, in more than 50 countries, with a budget of over $2 billion, with a huge portfolio of programs, which include the Global Disease Detection Program, Global Immunization, an influenza program, which largely WHO dictates what's going on, the malaria program, the field epidemiology training program, which I'll speak a little bit more about, and collaborations with the Department of Defense, and of course PEPFAR, the HIV/AIDS program. So we tend to promote health through strong partnerships by expanding the scope and depth of public health services both domestically and internationally. So we work with international health organizations such as WHO and PAHO. We work primarily with ministries of health and other health departments, NGOs and foundations, academic and scientific organizations, and public health associations and practicing professionals, and not least private industry, because we believe that we can make some contributions that other agencies cannot make. So let's look at a couple of examples. So which are linked, if you like, to diagnostics. First of all, the strengthening of capacity to conduct effective surveillance. And in order to conduct surveillance effectively, you need good tools. And it's often important to integrate multiple surveillance systems at the same time. Rosanna yesterday spoke about syndromic surveillance, and that's something I'm going to expand on a little bit in a couple of minutes. So the use of surveillance information and the flow of public health data, we can't be the world's laboratory, but we can help strengthen laboratories. And through the ASLM, for instance, in Africa, we're strengthening the capacity of laboratories to provide good surveillance data. The increased involvement of clinicians to emphasize community participation and to use data to respond to disease and other public health events. Ultimately, to improve compliance with international health regulations, and that's what the Global Health Initiative is all about, and we're one of the leading lights in that. Okay. So how do we interact with international partners? Our main partners are ministries of health. We have a history of provision of technical assistance and capacity building internationally. Our field epidemiology and lab training programs are partnerships with often local ministries of health and also universities, because often this leads to master's degrees in field epidemiology or laboratory science. And this is building capacity for countries to collect their data, to make sure that the laboratory data that's collected is accurate, and to use those data for public health decisions. PEPFAR funding is used to strengthen generic public health laboratory systems, and CDC has been a really key partner in the development of the African Society for Lab Medicine that you've heard about before. Just moving forward is the Global Health Initiative, which has been given such a kickstart with Ebola. Everybody now realizes that literally the worst diseases are just one or maybe two flights away from any part of the world. And so it's very important that we actually have integrated epidemiology, lab, policy development, and health communications in order to respond to these health threats. So, let's start looking at examples of how we collaborate in diagnostics. Well, there's our international laboratory branch in the PEPFAR group has developed literally an incidence assay for HIV, which is now being commercially made available by 3 different companies. The great thing is that there will be competition, and this should drive the price down. And remember, this is an incidence assay. It's not a just detection. One is trying to find out recent infections to determine whether or not there's been an impact of an intervention. The development of a cryptococcal meningitis test. Once again, a collaboration with a company, and this is the detection of cryptococcal antigen in blood and in a very early stage, and therefore enabling patients to be put on suppressive oral therapy rather than waiting for detection of the antigen in the CNS and CSF, and in order to by which time amphotericin B intravenous therapy is required. I'll show you a little bit about development of dual syphilis tests, either treponemal, non-treponemal tests, or treponemal HIV tests. The exploitation of a Luminex platform for detection of neglected tropical diseases and vaccination markers, and I'll show you that in a second. The development of real-time PCR for STI surveillance in order to support syndromic management in developing countries, and collaboration to develop highly multiplexed assays for pneumonia surveillance and other indications as well. So the one thing that you can see from most of these is that we're looking at multi-pathogen testing. So because it provides a more comprehensive picture, you can look for co-infections, and a carriage state, etc. It saves time with simultaneous testing of a variety of microbes. So you can perhaps look at bacterial versus fungal versus parasitic, all in one assay. And it conserves specimen volume and cost. You only need one specimen, and you can perform a number of investigations at once, and improves patient and public health response time. So, keeping it simple, this is the dual treponemal, non-treponemal lateral flow test, which actually has improved and reduced the amount of overtreatment as a result of being able just to detect lifetime exposure to syphilis, and more recently the non-venereal treponematoses of yaws in the South Pacific and in West Africa. So with this particular test, a negative test has just the control line. A confirmed positive test has 3 lines, a treponemal, a non-treponemal, and the control line. A biological false positive actually just has a non-treponemal line and a control line, and probably a previously treated case would have a treponemal line and a control line. So that's now widely used as a cornerstone for yaws eradication worldwide. This is something completely different. These are blood spots which are being collected here, and this was during a yaws campaign. And you see these little blebs which are covered in blood. And you can literally snip these off, elute the blood out of them, and you can perform a Luminex assay. And this is literally an assay which you can get an antibody snapshot of what's going on in a community of children, for instance, under 14 years of age. You can see whether they have a problem with lymphatic filariasis, whether they have a problem with trachoma, schistosomiasis. You can make an assessment of their vaccination status. You can look for giardiasis or ETEC or amoebiasis. Obviously, it's not a definitive personal diagnostic test, but it gives you a snapshot, and you can do all those investigations on the elution of one of those little blebs on the side. So this is true multiplexing, and it's being more and more used in order to look at the kind of problems that communities face. Just to give you some idea, this was an evaluation of the Luminex treponemal test versus the gold standard, which in this case was the TPPA, the particle agglutination test. And you can see the sensitivity was 98.6 and specificity 98% against that gold standard. The other nice thing is we can work with industry to augment the assays that we have, and this is the FTA card. Now, this is just it's just literally like a piece of blotting paper, but it's impregnated with chemicals which inactivate proteins, inactivate bugs, but maintain the integrity of nucleic acids. And so when you apply a swab to it from any kind of lesion, you can allow it to dry at room temperature, and you can then put it in the post, literally. It's of no risk to anybody, and we've used this in remote areas. And then when it gets to the lab, we punch out a little punch from the card, elute it, and perform real-time PCR on it, and the results are just about equivalent to actually taking the swab, putting it in the fluid, and running the test on the spot. So that makes surveillance so much easier as a result. I want to finish by showing how sophisticated these multiplex assays can be. And this is the TACMAN array card, which was previously known as TILDA. It's a real-time PCR system for rapid detection of multiple agents, and it was developed collaboratively by 3 respiratory divisions at CDC with Life Technologies. It incorporates CDC-designed assays, and it was customized for public health needs. So it goes onto a commercial card and uses a Life Technologies instrument, and it was initially used for outbreak investigations of unknown etiology, including during the 2009 flu epidemic. So it's now routinely used by CDC's Unexplained Respiratory Disease Outbreak Working Group to find out what the heck is going on, particularly with childhood pneumonias. Literally, you can simultaneously test 6 patient specimens for 48 different targets. It has a simple setup, low contamination risk, and you have preloaded primers and probes, and you need a minimal specimen volume of 50 microliters of total nucleic acid extract in order to perform the test. And at the back, you're not going to be able to see exactly the kind of panel that one has, but it involves various viruses, bacteria, and and can be used for fungi as well. And this is the pathogen sushi list that we've developed so far. And you can see that it really encompasses a wide range of pathogens, and that's expanding literally by the day. And the big benefit is if you were using an individual PCR assays, the specimens required would be something like 240 microliters of extract, and the runtime is many hours. And with the TAC cards, the specimen uses the 50 microliters, and we're talking about far fewer hours in order to be able to perform the testing. And once again, this helps with syndromic approaches to, in this case, the treatment of pneumonia. So literally, we have the extraction, the inoculation of the cards, putting in the machine, and then the interpretation of the real-time assays. So it's been applied to outbreaks, to population-based surveillance studies, domestic and international, specialized studies, for instance, neonatal sepsis etiology in Asia, and there are other additional syndromic studies pending. And guess what? Hemorrhagic fevers are part of those plans. Surprise, surprise. So I think I've told you about the kind of range of activities that we have at CDC and how we can work with industry and with regulatory authorities in order to provide the data that's required to give us good, reliable tests for, especially in resource-poor environments. Thank you very much.
Eu acho que foi uma excelente mesa de abertura, onde nós tivemos uma visão bastante ampla de algumas iniciativas regionais demonstradas pela doutora Rosana, a visão da OMS e, finalmente, a visão do CDC sobre esses aspectos de regulatórios, pré-qualificação e algumas novidades que estão no pipeline. Então, obrigado pela paciência, e nós vamos fazer a foto aqui mesmo, né, Carlos? Tá bom, então. Obrigado.